Hi everybody, welcome back. So in our last lecture, we were looking at the process of transcription, or how a DNA provides a template for mRNA to essentially extract the genetic code. And now what we're going to be doing is understanding how that mRNA helps create a protein. And that's really the process of translation, where we're going from this single-stranded mRNA to a ribosome to the creation of that single-strand protein, which then folds and creates that three-dimensional protein shape that we talked about previously. And really, the function of mRNA molecules is it's used as a template to assemble the corresponding amino acid sequence. Remember how proteins are a series of amino acid sequences, and that's what then creates our protein structure. And all of this process happens within the very important ribosome. So, as you probably remember from our organelle lectures, ribosomes are freely floating in the cytoplasm of the cell. So, what happens at this stage is the mRNA, again, your book is using this example of the antithrombin protein cycle, binds to the ribosome, and there are specific parts of the ribosome that do specific things, and I've included a really great visualization of both transcription and translation that really go through each of these steps specifically. So it would be great for you to watch that video and follow along with these lecture tapes as well. So once that mRNA has bound to the ribosome, it moves along the RNA and starts to read the groups of 3 nucleotides. So when we are talking about protein synthesis and protein translation, we're always going to be talking about these groups of 3 nucleotides. And that's because 3 nucleotides are what we call a codon. And these 3 nucleotide sequences, or this codon, specifies a particular amino acid, remembering that a protein is made up of amino acid chains. So let's look at a broad visualization of this. So in the ribosome, we have our mRNA that is bound, and as the ribosome moves along this mRNA strand, these sequences of freely floating tRNA, or what we call translation RNA. I apologize. I mean transfer RNA. So we have a messenger RNA, and then we have transfer RNA. And these transfer RNA are 3 or 1 codon units, and attached to each of these codons are the corresponding amino acids. So the ribosome is essentially taking this tRNA and connecting it with the appropriate 3 codon sequence. And above, you can see this growing amino acid chain that will eventually become a protein. So we're going to zoom in. Again, a lot of zooming in and zooming out in this lecture. But bear with me. At the end, there will be a protein. So let's zoom in on this idea of transfer RNA, or tRNA. They're carrying these amino acid sequences to the mRNA ribosome. And the ribosome is essentially finding the matching codon. So we have an anticodon here, and then we have the codon down here. And as these tRNAs are attached from the ribosome, we're creating this growing peptide chain. And this is what essentially the DNA originally was coding for, because from that DNA instruction manual, we're able to create a messenger RNA that contains the code, and then you have essentially tRNA coming in with the anticode, which creates this growing peptide chain, which creates a protein. So when the correct tRNA is in place, the specified amino acid, again, in this case we have sertraline, is added to this growing chain. And then the ribosome moves on to the next codon or the next 3-unit sequence so it can find the specific anticodon of the tRNA. And again, that chain just continues to grow as the ribosome moves up the mRNA. And once this completed amino acid chain is finished, or the ribosome finishes with the mRNA strand, the completed amino acid chain detaches and begins to fold into its 3D shape. And that's where you're going to get your completed protein. And in this case, we have, again, the antithrombin protein. So let's think a little bit specifically about that genetic code. So we went from a DNA sequence to a mRNA sequence to a tRNA sequence, and that encodes for a specific protein. But within this genetic code, it's good to understand that there's a set of rules of relating particular RNA codons to particular amino acids. And it's redundant and universal. So this is just an example of the different codons that are possible and what they can code for. So for example, all 4 of these sequences all code for the same amino acid. So that's where you get redundancy. But there's also very specific codons as well. For example, AUG is what we call the start codon. So this is where a proton sequence starts, is whenever you have this AUG codon. Simultaneously, we also have stop codons. So we can tell the polymerase to stop at specific coding sequences. And that's where these stop codons are important. So the main thing to take away from this, I think a lot of professors like to go through and have you start with a DNA sequence and build up a protein. And I'm less interested in you guys being able to do that, but rather just to understand that within the genetic code, there are redundancies and there are things that are universal. So there are things that will always code for something, and there are also contingencies that code for multiple things. And that this is ultimately what makes up a specific trait and what creates a specific protein in the cell. So again, this is really the process that is allowing us to create the basic units of life. Now, how does this all relate back to that mRNA vaccine? So when we last left off, we said, okay, we're able to create this mRNA that's going to code for a specific protein. But what good is that if we just inject an mRNA? What does that mean? Well, interestingly, when you inject an mRNA, our cells will actually translate that protein into proteins, which then will create antibodies. So the cell will start to express this spike protein, And our bodies will go and say, oh, what is that? That's strange and different, even though it's missing the virus itself. It's just the expression of this spike protein. Our bodies can start to create antibodies to this specific protein. So if we become infected with the real virus, in theory, if you've been inoculated with this vaccine, your body will already have these important antibodies and T cells saying, oh, I recognize this protein from this original mRNA sequence, and I'll be able to defend against it and not make my body so sick. And I've included a couple other videos that go over this process a little bit more specifically. But what's important to take away from here is that mRNA vaccines are a very exciting field of research, and it's a great way for our bodies to start to recognize foreign agents, such as this spike protein from the coronavirus. And the only reason that this whole process is made possible is because our bodies are constantly going through this process of transcription and translation. So in summary, genes provide the instructions to make proteins, and this is a process called gene expression. And proteins are simply folded chains of amino acids, and the important key here is that they are folded. Because only once the amino acids become folded and interact with its side chains and its component parts do you actually create a protein that is then functional. And the amino acid sequences, so the order of the amino acids, determine the shape and function of those proteins. And any change in that DNA sequence of a gene can change the corresponding amino acid sequence and therefore the function of a protein. And also, we also learned that every gene has two parts. There's a coding sequence and a regulatory sequence, and how those are related to transcription and translation. And lastly, we learned that gene expression occurs in two stages, transcription and translation, and we went through how those processes interact. So I know that was sort of a long lecture. Good job. I know this is a little bit complicated, but make sure you watch those additional videos that show the process of transcription and translation. Follow along with your notes and get comfortable with how our bodies synthesize proteins. Okay, thanks so much, and I'll see you next time. Bye.